Hopefully, at this point, you are all saying to yourself, well, protein synthesis sounds great, transcription and translation sound cool, but how do you know the order of these amino acids? And there is a really cool code that we get to break down during translation. And that code, that genetic code, um, it indicates and tells the order of the amino acids that make up a particular protein. So, we're going to talk about that code, how to read it, how to decipher it in this video. So, here we have this concept of translation. And if you recall, we have our tRNA little person here. And this down here, this is our mRNA. And on our mRNA is the codon. And in this case, AGU is the codon on the mRNA in this example. And that codon determines this thing at the top here. This is the amino acid. And I'm just going to put an AA here for amino acid. And SER probably doesn't have a whole lot of meaning to you right now, and that's okay, because there is like a little cheat sheet in biology. And this is our little cheat sheet for telling us what amino acid the tRNA gets to bring. So in this slide, the codon reads AGU. And if we go to our wheel and we find A, and then we find G, notice how we went out to the next layer. So I went from A to G, and then that last letter was U. So AGU, we work from the inside out. Always work from the inside out on the wheel. And we're not going to do too much of this right now, but I just wanted to kind of get you familiar with the idea that in reading and deciphering and cracking this code, it starts with the triplet code, the codon that is on the mRNA. Okay, and that triplet code is really it's a word kind of, and that word is the amino acid. We read the code, and that code tells us which amino acid comes next. And this code is pretty much universal, like all living things use it, which is really, really neat. So, for example, this would be like an mRNA sequence, AUG, and then you could have you have stop sequences. So this is a start sequence, AUG. Again, it's read in groups of 3, so we have to divide up our mRNA into groups of 3. And we have some stop signals too, which is kind of neat in this little language that we're talking about. So again, we have, and I'll highlight this in the next, yep, here we go. So we have 1, 2, 3 bases in a codon, and those 3 bases give us 1 amino acid. So 1, 2, 3 bases on the mRNA give us 1 amino acid, and that amino acid is carried or transferred by tRNA. Here we have UAC. UAC is the codon for the amino acid TYR. Again, TYR probably doesn't have a whole lot of meaning to you, and that's okay, because that's why we have our handy dandy little wheel that we can go back to. And I will attach a wheel for, you're welcome to use it as you want to try to do this step here, this reading or cracking the code. But if we have UAC, if I go to my wheel, again, we have a U, and then we have to go out to the next layer, A, and then C, and that tells us the amino acid tyrosine. Do I have this wheel memorized? No, it's about knowing how to use it. So again, you work from the inside out, and that's how you essentially read and crack the code. This is how we get the order of those amino acids in our protein. And again, if we're looking at this, these are all the different amino acids around the outside here. It goes all the way around. And these are the mRNA bases in the middle. And we read the code, again, in groups of 3. So 3 RNA bases for every 1 amino acid. Okay, so we call that code a triplet code. And that information is here. Again, I will post this presentation as well. We talked about working from the inside out on the mRNA and reading that mRNA codon in groups of 3. And I really hope this is helpful. We stop the polypeptide or the protein with what's called a stop codon. And these are our 3 stop codons. It's programmed in the language, which is really, really cool. And then that mRNA gets released and the cell kind of goes about its business. So again, here's the big picture. We have DNA in here, which this is the nucleus. The DNA message is written down in mRNA. The mRNA travels out of the nucleus. It finds a ribosome here, and that message is translated. We have our tRNA bringing over the amino acids and matching those, matching up with the right group of 3 or codon. So again, this codon in the mRNA corresponds to the amino acid, and that is how we read the code. That's how we read the genetic code, and that is how we get to make or synthesize a protein. And I hope you found all this interesting. Again, please watch all the videos in order,